Good morning, everyone. I'll continue my previous lecture where we talked about the microbial diversity and systematic classification. So, in my last lecture, we talked about mostly morphological and conventional method of classification. But today, we'll talk on the molecular aspect and the molecular evolution in the microbial diversity. As we discussed yesterday on the microbial diversity, where we talked about the systematic classifications and identification using the morphological characters, which are not ultimate because through these characteristic features, it is difficult for identifying the specific species or even the different diversity within the species as well, like strains and types and all. So, the method which we discussed today or the molecular evolution in the microbial diversity will help you out to understand how the microbial diversity can be fully understood through molecular aspects. So, the first is DNA based techniques. In DNA based techniques, which transform the whole taxonomy by using the different modern methods using the nucleic acid. So, the first we'll talk about the GC content. Now, the two microorganisms having a two different genome have a different GC content. So, having the GC content estimation, percent GC content of one organisms and percent GC content of another organisms will definitely help to differentiate these two organisms separately. So, this is one aspect where the GC content, for example, Mycobacterium tuberculosis, for example, E. coli, Klebsiella pneumonia, Pseudomonas, and several other microorganisms which belongs to different categories. Every organism has a different GC content in the genome. So, the genome diversity will basically help to understand the microbial diversity. Similarly, DNA hybridization. In DNA hybridization techniques, we measure the genetic similarity, how the two DNA or two nucleic acid or two genome are similar within the two individuals, which we are looking if they are from the same group or same class. Now, in the case of hybridization, the 90% homology or 99% homology will tell you that this organisms belong to the category to which we are looking or to which we are placing that organisms in that group. As yesterday we talked about the stability and predictability, which is one of the two important component or the requirement for the classifications, where if the stability and the predictability is not there in the classification, then probably your classification is failed. So, GC content and hybridization, DNA hybridization, or we call the nucleic acid hybridization, probably help you to understand this, to maintain the stability and predictability. Then you have a 16S RNA sequencing. The 16S RNA sequencing is the ultimate technique where you can identify a particular species. Now, Carl Woese, he was a scientist who first identified the ribosomal RNA sequence. These ribosomal RNA sequences are highly conserved sequences, and every individual within the species are supposed to have the same RNA sequences, ribosomal RNA sequences, which are about 800 to 1 kb base pair sequences. Now, the universally conserved gene of ribosomal RNA will help you to identify different individuals, different organisms belonging to particular groups. In domain, in bacteria, archaea, eukarya, and all kind of individuals. Now, these molecular techniques enable this construction of a phylogenetic tree. Now, the phylogenetic tree can be constructed through 16S RNA sequence data, or we call the 16S ribosome RNA data. Well, the cultivation independent approach, for example, many microorganisms, they can be cultivated, they can be cultured. And majority of the microorganisms, if I say more than 90% or even more, microbial world, they are non-culturable. You cannot cultivate these microorganisms, you cannot grow these microorganisms in a media, in a culture, in the lab. So, how do you know these microorganisms? This is the big challenging question. What are the approaches to take to classify, to identify these non-culturable or non-cultivatable organisms? So, organisms which cannot be cultivated or which cannot be regrow in a in the laboratory through artificial culture media can also be identified through some techniques. So what are these techniques? Unlike the earlier, we grow these microorganisms in the culture, and then after growing in the culture, we take these microorganisms to know the RDNA sequencing, to get the DNA for hybridizations, to look into the similarity between two individuals, as well as we go for GC content through sequencing. But those which cannot be cultured, which cannot be visible to you in a particular site, for example, clinical isolates, environmental isolates, water bodies isolates, poultry isolates. So these isolates cannot be, or even the soil isolates in case of various bacteria which are good for the nitrogenous compound providing in the plant. So for that we have to go for the metagenomics and metatranscriptomics. Now what is metagenomics? Metagenomics means you have a direct sample without isolating the organisms from these samples. You go for DNA sequencing. Now the direct sequence of the environmental DNA sample, for example, if you take the water sample or if you take any clinical sample and you just get the DNA out of it. So you can ask a question, how can you isolate the individual's organism's DNA? And when these multiple organisms present in the environment, DNA mixed together, how would you separate them? How would you characterize them? So this is a whole area to understand what we call the metagenomics. Metagenomics are basically divided into two categories. For identification and classification purpose, metagenomics are divided into two purposes. One is called 16S RNA metagenomics. Then we have comprehensive whole genome metagenomics. Now, for the purpose of identification from the individuals from the multiple site, you need not to go for the comprehensive metagenomics, but it is always good that to go for 16S RNA metagenomes. And this 16S RNA metagenomes will uncover the what kind of a bacteria or bacterial populations or population present in the particular sample, water samples, clinical sample, or any other samples, even soil ecosystem. So this is one of the best technique today through whole genome metagenomic sequences. Now, metatranscriptomics is another area. In the metatranscriptomics, the analysis of the environmental RNA, transcriptomics, when I say, it means it tells you about the transcripts identification, that is the gene expression profile, gene expression pattern in the natural communities, like it can reveal the microbes and metabolically active microbes under specific conditions. So if you are going for transcriptomics of a particular individual ecosystem, for example, from soil and from gut metagenome or from oral metagenome, oral microbiome, then you can have an idea about how these microbial world in the particular ecosystem or particular niche is basically providing the conducive environment, conditions to express the gene. So transcriptomics or metatranscriptomics is the area where the gene expression can be identified. So by identifying the gene expression, you can again have a systematic classification of the bacteria and microbial world, looking into what kind of a gene or what sort of genes are being expressed and what sort of genes are not being expressed. For example, I can take an example here in case of drug-resistant genes. Now, in the drug-resistant gene, you can see the vast varieties of the genes are present in the microorganisms, in different microorganisms and different settings. Now, when you go for metatranscriptomics, you will identify the expression of certain genes, and you can look into which environment, which ecosystem help you to know to have a particular kind of a gene carrying bacteria. So this is one advantage of this metatranscriptomics at the level of gene expression. In the community profiling, the environmental DNA sequencing enable comprehensive community profile, identifying specific composition, relative abundance, and ecological relationship within complex microbial ecosystem. So these are the areas where we can explore the different microbial world in this. Just take an example of case study. Mushroom composed microbial community. Studies demonstrate how cultivation independent approaches reveal succession dynamics during the decomposition. It means the whole mushroom compost which are formed as a decomposition in the soil or in the particular niches in particular ecosystem, how it decompose and what is the decomposition dynamics, what kind of a bacteria are involved in that, and how these bacteria can be identified. So all these things can be identified and can be used. So it is one of the benefit of such kind of studies that you can know which environment, which ecosystem has what kind of a bacterial population, what kind of a microbial population, so that you can use that ecosystem for isolating such kind of microorganisms for the benefit. And the same is fit for the purification of the wine and purification of the several fermentation food which ferment and which give you some good quality foods through fermentation. So you can identifying these strains. For example, Saccharomyces cerevisiae strain, I identified the best strain to produce the best quality wine. So from that environment, you can easily isolate that, and you can use that environmental isolate for production of the good quality wine. So these are the advantages of this kind of a study to look into diversity of microbial world and the systematic classification. Now, statistical approaches to diversity analysis. How would you analyze the diversity? What are the different approaches to analyze the diversity? And again, I'm repeating the same thing, that what is the significance of classification? Until you characterize your organism on a very unique characters, it is difficult for you to keep them onto different orders. And until you keep them on different orders, like hierarchical orders, like a domain, kingdom, order, class, a family, genus, species, and so on, then it would be very difficult for you to pick up a very specific required microorganisms for any cause, whether for research study or whether for some benefit, for some food production, fermentations, and so on. So there are some statistical approaches are defined here to analyze the diversity. Richness estimation. Suppose if you go to the whole genome sequencing or metagenomics, so in that case, the richness is one of the things that you have to look into, the richness of or abundance of the microorganisms. Now, once you identify the abundance and the richness, this will help you to look into the kind of a diversified microbial world present in that particular environment. You can also change certain conditions. For example, you can change certain parameters like environmental parameters, some media conditions, and you can look into the dynamics of the diversity taking place in that particular populations. Evenness analysis, for example, statistical measures of evenness such as Shannon and Simpson indices. These are little advanced technology which probably it is difficult to understand because the whole lecture is required to even talk about the richness and evenness analysis, but just to understand how these techniques, statistical techniques, help you out to identify particular microorganisms in an environment. Ecotype simulations, again, ecotype simulation is an advanced computational tool which can help you to understand and to simulate particular ecologically distinct population of a microorganisms which represent the separate species and subspecies. So you can also categorize the species level and beyond the species level with the similarity thresholds to recognize the evolutionary and ecological units. Now, these statistical frameworks and these statistical diversity basically help us to precisely identify a particular microorganisms and try to unrelate them from the other microorganisms present in that particular niches, in that particular ecosystem. The modern phylogenetic tree reveals the true diversity of microbial life. Now, the phylogenetic tree is something related to, kind of a pedigree records. If you look into like a family, you have a pedigree record, your, grandfathers and grandfathers and great-grandfathers, and then your fathers and son, and so on and so forth. Like this, if you look into the the whole pedigree record, genetic diversity, from generation to generation to generation to generation, you find similarity and variation through the common phenomenon of recombination, as we all know. Again, I'm not going to talk about what the recombination is, but by exchanging the genetic material from generation to generation and transforming material from generation to another generation, make the differences or make the variation among the species, and that make the diversity in the related or unrelated organisms. So phylogenetic analysis or phylogenetic tree will be the best tool, or best rather platform to understand the diversity and to also display the diversity, where the major lineages represent the entirely uncultured organisms as well. So if you go through the data generated through metagenomics, and this data of metagenomics will analyzed through phylogenetic analysis, through different softwares, computational based softwares, will give you the clear-cut picture where your microorganisms are present and how you can differentiate these two on the basis of the phylogenetic tree. Now, each branch represents the millions of years of evolution. So it takes like number of years and years to evolve a species or to evolve individuals from one type to another type. And these gaps highlights how much we still have to discover. Now, this is the area where a lot of genetics and mutations are involved, which are the normal or the common phenomenon. We call the spontaneous mutations phenomenon. We are not talking about the induced mutations. And these mutations will lead to through many ways, as recombination is one of them. Then we have transposons, then we have jumping genes and IS elements, integrons, even the phage through horizontal gene transfer. Probably in next few slides we'll discuss also what the horizontal gene transfer is, how the genes are being transferred and make the variations. But these variations basically ultimately has to be judged, to be tested, and to be identified through these methods, and to give you the clear pictures how these are varying through these things. And the modern microbial systematic classification, what are the different techniques? In the modern era, we have a different techniques which emerged and which came out to reduce the time to analyze to produce the data, to generate the data, and to interpret the data, as well as the precision is also increased. Polyphasic taxonomy, integrating multiple data types. So we have a multiple domain, for example, phenotypic data. So on the basis of the phenotypic data, which is traditional morphological, physiological, and biochemical characteristics, as we discussed in my last lectures earlier, talked on the diversity and microbial diversity and systematic classification, where we talk about the methods which are related to their physiology, nutrition requirement, culture, metabolic conditions, all comes under the category of the phenotypic data. So to comprise the phenotypic data, compare the phenotypic data, variation among the individuals, enumerate them, and to compare the relatedness and unrelatedness through these two generations, through these two different individuals, right? So traditional morphological, physiological, biochemical characteristic features, it continues to provide the valuable taxonomic informations, especially for culturable microorganisms and practical identification purpose. Now, if the organisms which cannot be cultured, the phenotypic data can't be used, obviously, right? For that, the genotypic information is required. DNA sequencing, genotypic content. When I say genotypic content means what different contents present in the genome. There's a vast varieties of the genome diversity that we talk about. For example, the type of genome, gene, their expression, transcriptomics through transcriptomics, the gene structures, presence of different integrons, jumping genes, and so on. So the genetic markers offers the objective measures of the relatedness. Now these markers can be studied through the relatedness, how they are related, how many markers you identify, how these markers are related to the other species, or how these markers are related to the species number 1, species number 2, and so on. forming the backbone of the modern taxonomic decision. So the modern method of genotype information is very, very important to identify precisely a particular individual. Ecological context. In case of ecological context, you talk about what is the ecology of these microorganisms, where they grow, what is the habitat, how they are associated, and important dimensions to the taxonomic classification. So under one category of classification, the organisms are having the habitat, say for example, 1. In other case, they have a different habitat. So how these two different organisms having two different habitats are related or unrelated? Again, you can correlate and associate with several other characters. And this recognizes the ecological and evolutionary background of these individuals. The phylogenetic analysis, evolutionary relationship inferred from molecular data, that is the data generated through genetics, generated through sequencing, generated through transcriptomics and so on. And that data provides the theoretical framework for understanding how species are related and how they should be classified, right? The polyphasic taxonomic represent the current gold standard in microbiological systematic classification, combining the multiple lines of evidences based on phenotypic data, based on genotypic data, based on ecological data, based on their phylogenetic analysis, or relatedness, or similarity, or relatedness, or variation. So all these data together give you a polyphasic taxonomy, which is the clearly identifying a particular species or individual in that case. And the genomics and the comparative genomics. What does the genomics and comparative genomics means? First, try to understand the genomics. Genomics is the study where you talk about the whole genome. When I say what is genome, genome is the n number of chromosome of an individual. For example, if you talk about the human chromosome, right? What is the n number of chromosome in human, right? So there are 23 pair of chromosomes. It means 23 chromosomes, that is n number, in human is the genome. Now, it has a complete information about the gene and the alleles which are present in that genome, right? So, to look into the whole genome analysis and we compare that genome to the other individual genome, give you a clear-cut picture how it varies. What is the percent similarity? For example, if you talk about the E. coli and if you talk about the Klebsiella pneumoniae, both belongs to same family, Enterobacterial or Enterobacteriaceae. Now, if you look into the character, primarily the characters are similar. Phenotypic characters, as well as the genotypic characters, as well as ecological characters, as well as some phylogenetic characters are similar. But when you go more into detail about the genome, you find the relatedness, you find the variation, you find the genes, you find the gene number, you find the gene size, and you find various other markers which are present in that, which make you differentiate these two individuals at two different genera or two different species later on. Similarly, within the same species, for example, if you talk about the Escherichia coli, or if you talk about the Klebsiella pneumoniae, if you talk about the Pseudomonas aeruginosa, Pseudomonas putida, so these are the two different species. Till the Pseudomonas genera, you find a lot of similarity in the characters. And so you say, okay, this is the Pseudomonas, but still you are not sure whether this is Pseudomonas aeruginosa or it is Pseudomonas putida. So to know the Pseudomonas aeruginosa and Pseudomonas putida, to differentiate these two, you need to have some different molecular method. So this is how the genomics and comparative genomics requires. A species boundary refinement. Whole genome sequencing provides unprecedented resolution for defining species boundaries. Where does the species stop? Where does the species start? So revealing the cryptic diversity, right, and clarifying the relationship between closely related organisms. It means, for example, closely related organisms, in which context you are talking about, whether at the species level, genera level, class level, or order level. So this you have to look into. Through genome-wide comparisons, right, the whole genome comparison. Evolutionary history. Comparative genomics reveals patterns of horizontal gene transfer. Genes are transferred through conjugation, through transformations, through transductions, genome rearrangements, and evolutionary pressure, right? And and that shaped the microbial lineage over geological time scale. Over a period of time, maybe hundreds of years and thousands of years, it keeps changing. Functional prediction. How do you predict functionally? What are the different functions of these microorganisms? Genomic analysis also predict the metabolic capabilities. What are the different metabolites which are produced? Ecological roles. Where does it live? What is the habitat? And what is the biotechnological potential? How it is being used for the biotechnological production? Even those that cannot be cultured in laboratory condition. Even those microorganisms which cannot be cultured in the laboratory. So you can judge, you can check through metagenome analysis. Now, what are the challenges in defining the microbial species? Horizontal gene transfer, right? Then extensive genetic exchange of the genetic material from one species to another species, and even across the species and across the genera it is possible, right? So the transfer through not vertical, not through sexual reproduction, but across the boundaries of the species and genera, genes can be transferred through various methods we call genetic methods, conjugation, transduction, transformations. And through these transformations, the lineages are changed, the microbial genome is changed, and so you can, on the basis of that, you can categorize them. Okay, this is having bacteria have this plasmid, this bacteria lack that plasmid. So on the basis of the presence and absence of that particular plasmid, which is being transferred through horizontal gene transfer, can be judged into two categories, and they can carry the different genes, different characters, different functions. Ecological specialization is ecological distinct population, that is ecotypes, may be genetically similar yet occupy different environmental conditions, right? It may be similar, but they can have a different habitat. They may be humid habitat or they may not be humid habitat. They may be dry habitat or they may not have a dry habitat, though they have a similarity. So this kind of a population variation or population variation can also be one of the challenge in defining the microbial species, because there's a lot of variation were found, and you have to sort out how they differ, what are the basis of the difference in these two different species on the basis of the ecological and or through transfer of the genes from one species. For example, if you say E. coli having the plasmid, cryptic plasmid. Now another E. coli strain may not have that cryptic plasmid, and the E. coli which is having the cryptic plasmid may have some gene which are not present in the other plasmid. So that have functionally different, right, phenotypically different, and they can be categorized into two different classes and two different categories at the hierarchy level. So these are some challenges where we have to face sometime. Now arbitrary threshold, the current species definition, that is depends on somewhat arbitrary genetic similarities cutoff point, like 97% 16 RNA identity or 98% or 99%. So you have to set a boundary, some line there, this similarity to be taken for this categorization, or this similarity, percent similarity to be taken for this categorizations, and so on and so forth. So then so you can have your own way of classification. Now, theoretical framework gaps, the lack of sometimes unified theoretical framework that integrate genetics, ecological, evolutionary criteria, as we discussed already. Different research communities or researchers, they often apply different standard leading to non-culturable or bacteria, how to nomenclature to be set, or what kind of a nomenclature to be set for that, and how these species names to be given, and how genera names to be given. So all these things to be taken into consideration based on different region, different ecosystem, different or diversity, mode of diversity of this microbial world. So there's a lot of challenges to be faced in order to define the species. I just take an example. Suppose you identify a species in India with all like hundreds of characters to be given that species. Okay, this is defined as a species X of E. coli. Now, when you take this E. coli, a new species we called as E. coli X or some different type of E. coli, same species with the same character identified, say for example in the United States, right, and having same character, 99.9% same character, but there might be a chance that name may be given in different way. So we can have some different nomenclature for that. Now, in order to give the uniform nomenclature, this is important that to look into the same kind of species characters and try to take these characters 99.9% similar to identify that species worldwide or globally. Now, to explore the genome, the modern genomics revealed the intricate patterns of the microbial genetics diversity. And this through genomics and metagenomics we already have talked about. Now, thank you very much for this. And now we have another lectures on the applications and implication will start soon.